president of the National Foundation of Swallowing Disorders. I want to thank all of you for joining us this evening. For those of you who haven't joined in previous TED Talks, a few ground rules. You will be muted during the presentation. If you have questions, there's a question box, and you can enter a question at any time. Michael and Jonathan have shared their talk for 30 minutes, so there will be plenty of time for a dialogue with them. This will be recorded and available for free on our website, and there are no ASHA CEUs offered for these presentations. I would like to recognize Bracco Diagnostics for their unrestricted educational grant. It allows us to provide these programs without charge. Many of you have donated tonight, and I want to thank you for those donations. It helps continue with our mission. I would also like to thank our educational sponsors, Simply Fair Mobility, Marriage and CF, Prayer Rehab, and Pathway Muir. They also help to continue to support our mission. A little background on tonight's TED Talk. This program goes back over a decade. This is a picture of a group brought together by the foundation at the UC Davis Institute for Regenerative Cures. That's me in the foreground. Sonia Blue is in the foreground. Dan Alta, the head of the IOC, also in the foreground. In the upper left is Dr. Belafsky, and in the far right is Michael Donovan, and his sister, Pat Jelly, is also in the picture. But in 2013, Peter asked if the foundation was interested in trying to raise funds for a ton stem cell initiative in mouse models. And this group of people, along with primarily people tied to Dr. Belafsky, raised $40,000 for a stem cell trial in mouse models. And that provided those funds help provide the work upon which what you're about to hear about tonight is based. So this has been going on for, again, over a decade under the guidance of Dr. Belafsky. Next slide, please. I'm not going to read this, but it was, I'll let you read it while I talk to it. It was in Paleo News, and it talks about the challenges and the toxicity that many long-term head and neck cancer patients face. And I just came across this about 2 or 3 weeks ago. And having just gone through several infections, including a minor pneumonia, much of it tied to having a very weak tongue. I thought it was a very timely summary of an issue that MyoSite and Dr. Belafsky and team are trying to solve. Next slide, please. At this time, I would like to introduce you to the presenters. Dr. Michael Griffin has worked for MyoSite for the past 3 years. His undergraduate work was, or he has an undergraduate degree from the University of Pittsburgh and a PhD degree. Michael was kind enough to provide me with a tour of the MyoSite facility about a year and a half ago. And it is a world-class facility. Dr. Jonathan Anderson is a translational stem cell researcher. And I think it's important to define translational research. And what that means is we're looking to bring to market either in clinical trials or approved therapies within a 5-year period. So with that, I will turn this over to Michael and Jonathan. Thank you both for volunteering your time this evening. And I'm going to shoot for a while and turn it over to you.
Thank you very much, Ed, and thank you and the foundation for giving us this opportunity to talk this evening. And thank all of you attendees for being here today and for our talk. Just want to verify, Jonathan, you can see my slides. All right, fantastic. Then we'll move right forward. So this talk is on exploring. Give me a second. Exploring tongue muscle regeneration for dysphagia. Now, before we really get into the main topic of dysphagia, I do want to explore one concept of movement in humans and machines, and looking into what the differences are. Now, if we were to look at a chess game, so say it's how robots move and how they're able to move these pieces on the chessboard. It starts with actuators. So actuators are typically controlled by electricity or pressure, and it's this stimulus that causes the motor to spin and create these movements. It's this combination of the stimulus and the movements of the actuator that allow for these strong but also precise movements of the robotic arm. And that also, the actuators then control the servos. The servos are then precisely rotated and allow for that exact location and strength to do something such as complex as assemble a car, or something as precise as playing a game of chess, like we're using in our example here. Now, looking at the other side of the board, we look at the human player. The human player uses muscle fibers. These fibers extend and retract in order to make these fine motor movements of the fingers and the arm to pick up the pieces and place them on the board. In this case, the communication is coming from a nerve, which with that proper communication allows the muscle structure and to create these intricate movements. And that signal is coming from the brain. So the nerves then communicate that signal. The brain sends that signal to the nerves, which are then communicated to the neuromuscular junction, allowing for the muscle to know and to act, whether to contract or relax in order to complete the function. So when looking at these movements, whether it's the stimulus, the electrical signal to the actuator to the servo, or the brain to the nerves to the muscles, it's really not that much different in terms of the movement and how that actually functions. But the obvious difference comes when we break. When robots are broken, they require someone to come in and repair them. Whether it's the servo, the actuator, or another piece of that mechanical arm, someone needs to come in and fix it. However, in the case of humans, we have the ability to repair our muscles. We don't require, obviously we have doctors who can assist in this process and help aid with new technologies, but we have this innate ability to repair our own muscles, unlike the robot and machines. So this is pretty intuitive. I understand a pretty basic concept of how we play chess, how a robot could play chess, and then obviously what happens when either a robot or a human breaks. But have you ever wondered how we can do this? How we can go from a state of exercising and whereas these small micro tears or a larger injury with a larger tear of the muscle, how we can go from a state of being broken to some level of repair, whether it's coming back stronger or basically just coming in with some level of restoration of that muscle tissue after the injury. Now, in order to explore this, in order to find the answer, we have to go all the way back to the beginning. The beginning really being those early stages of development that we go through as humans. So starting with fertilization all the way through those embryonic stages and into this proliferation of cells. How are these cells governing this process? It starts with this concept of stem cells, where the stem cell, which is really this non-specialized cell, it can become anything. It itself does nothing, but it can replicate itself. It can go one of two ways: replicate itself, creating more stem cells, or it can go down this differentiation pathway and become specialized. And that specialization can allow it to become any type of cell type that forms us, whether that's blood cells, neurons, or in this case for this talk, and as we'll begin talking about dysphagia, when I hand it over to Jonathan, muscle cells, which we will touch on in just a moment. And I will attempt to describe the central dogma of molecular biology on this one slide, and how this cell fate is modulated by this process of gene expression. So this is really the foundations of molecular biology. If you remember back to your biology classes, we're made up of our DNA. So our cells have this DNA in them, and the DNA codes for is transcribed and codes for our genes. And these genes create really who we are as a person and what our cells are functioning to do. So the DNA is transcribed. It's transcribed into RNA, and the RNA is then translated into proteins. These proteins and transcription factors really govern how our cells operate and how our cells function and process and go about their processes. Now, in the case of this developmental pathway and how we get from this stem cell that can become anything to muscle cells, it goes through this proliferation and this proliferation of these transcription factors. And as the transcription factors become abundant and these proteins are abundant, specifically PAX7 here, and again, this is a very brief overview, not the complete picture, but something that is representative of what's going on in our development and in our regenerative processes. The stem cells can become satellite cells, and these satellite cells then in turn become muscle cells. But the satellite cell is this kind of intermediate cell phase. And what it is essentially it's a intermediate muscle cell, but it's not quite a functional muscle cell. And so what that means is it is, in a way, this regenerative mechanism without itself being fully developed into a muscle cell. And as it goes down this path from satellite cell to muscle cell, it continues to proliferate. This continued developing and becoming myofibers. So from this really early stage satellite cell that's beginning the muscle cell phase, all the way through myoblasts, MyoSites, which is where Cook MyoSite really has derived its name, myotubes, and then eventually into functional myofibers, creating those muscle fibers that allow us to move and function. And what that looks like, to utilize another analogy of the Russian nesting doll, is we start with these myosin and actin thin filaments that create the myofibers, and then it just keeps bundling and stacking on one another. Those myofibers bundle into the sarcolemma and then to the muscle fascicles, and eventually create the overall bundle of the skeletal muscle that is lumping all of these together and creating functional tissue that allows for these movements. But then looking into all this and how we go from that intermediate cell all the way to this functional skeletal muscle in development, what is the purpose? What is this point of the intermediate cell, and does it have a function as we are as adults and humans? So the point of this intermediate cell still is very much intact when we pass our development stages. When we exercise or get hurt, your muscle needs repaired. And so when that injury happens, the satellite cells or progenitor cells, they rush to the spot where the muscle is injured. And the reason they're called satellite cells is because they exist on the periphery of that muscle fiber. They rush to that spot and they respond to the cues, those transcription factors that occur due to the central dogma of molecular biology, of gene expression. Once they're at that site, the satellite cells that are there, they divide into muscle cells. They begin that differentiation pathway and become more mature muscle cells. They fuse with that existing muscle and repair the injury. And in addition, what the satellite cells do is they also create copies of themselves for the first time, or for the next time that the muscle gets injured. So in addition to creating this new tissue, this new muscle to repair the injury, they also create more copies of themselves so that they can be there to respond to injury as additional injury occurs. So what we're doing at Cook MyoSite, and what we have done over the past 20 years, is we are researching ways to harness this natural healing power of our muscle progenitor cells, or satellite cells, to help people with muscle disorders and injuries. And as Ed mentioned, we invited him over to our company headquarters in Pittsburgh, Pennsylvania, pictured here. This is our corporate offices as well as our manufacturing facility that we're building out here in Pittsburgh in order to create this technology and manufacture this technology for our clinical programs. And what the technology is ultimately is called, we term it autologous muscle-derived cells, or AMDC. This is the AMDC platform. And what that looks like is, knowing that we have these progenitor cells in our muscles, we take a biopsy from the patient's leg, specifically the quadricep, autologous being from the same patient. So the patient who is impacted in part of our clinical trials is also the one we take the biopsy from. We isolate out the progenitor cells from that tissue of the leg. And it's a small sample. It's smaller than the tip of your pinky that we take. And then we expand those cells in our manufacturing facility so that they eventually be injected back into the patient, whether it's for one of our pelvic health indications or for tongue dysphagia, as Jonathan will be touching specifically on next. And so those cells, with the idea that once they're in the injured tissue site, they respond to the cues of the injured tissue, and they proliferate and begin to integrate into that tissue, and hopefully restore that muscle function as it integrates and becomes, they become myofibers. And our company's goal through this platform technology of autologous muscle-derived cells is to make regenerative medicine a part of everyday medicine. And this is the mission of every single person who works at MyoSite, and something that we strive for every day through our work. And briefly, I do want to touch on our AMDC clinical programs, just to give context of what this is being investigated in. Our leading indication and what has been under development for the majority of the company's life is our female stress urinary incontinence indication. And in addition, we have 2 other indications that are in clinical development for underactive bladder and fecal incontinence for women with obstetric injuries. But then finally, for this talk, we are also investigating AMDC for tongue dysphagia. And Ed gave some background on this already, but I do want to touch on, it was about a decade ago, a little less than a decade ago, that Cook MyoSite was connected with Dr. Peter Belafsky on the potential clinical application of the AMDC technology for tongue dysphagia, specifically for tongue dysphagia for head and neck cancer survivors. And this has been a concept that he had been researching for a number of years prior to this engagement in this development pathway through preclinical work. And so at this point, I do want to hand it over. Now I've given some background on the technology and what the technology ultimately is. I do want to hand it over to Jonathan, who will be giving that background on where they have been with the preclinical work with the UC Davis group, but also where they have been with the initial clinical trial and where they're going with us, really as a team, UC Davis, us, and as well as UCSF now in the loop, where we're headed with this clinical program. So Jonathan, I will hand it over to you.
Great, thanks, Mike. Great talk. Mike is hard to follow. So I thought I'd just give a little bit of a background about the story of how did we get here. And the reason why I wanted to do that is because I feel like it's the coming together of a bunch of really great teams, and it's taken a lot of work, time, energy, and effort to get here. And so Ed mentioned at the beginning of the program that it was a grant from NFOSD that sparked the original research that eventually led to these clinical trials. But the story actually starts a few years prior to that with one of the big players in this picture that probably doesn't get as much credit because they kind of work behind the scenes, is this California Institute for Regenerative Medicine, or CIRM. And what CIRM is a state-funded stem cell funding agency that tries to bring these stem cell clinical trials into fruition here in California. So we're very lucky to be here in California and to have CIRM. And back in 2008, CIRM gave UC Davis a multi-million-dollar grant to build out the Institute for Regenerative Cures and to bring in Jan Nolta to be the director of this new institute to try to develop some of these stem cell therapies. So they sort of broke ground on that, so to speak, in 2008. And things were kind of up and going a couple years after that. And that's when Peter decided to go down and have a meeting with Jan. You know, a lot of you probably know Peter fairly well, big innovator in the field of dysphagia, and he's always trying to think of new ideas, new approaches. How can we do better? How can we do better? And it was with that sort of mindset that he came to talk to Jan to see if there may be this possibility of using stem cells to heal some of the damaged tissue in dysphagia patients. And Jan, to her credit, part of the reason why she's been so successful for so long is because she is a connector. She's always thinking of new ideas, new collaborations, connecting people, and that kind of plays out in the story too. So Jan and Peter kind of got together. They arced out on paper, how might we approach going from this cool idea of using stem cells to treat dysphagia, and they kind of laid out a roadmap. And it was in 2013 that roadmap got off the ground with the funding from NFOSD. So none of this would have taken place without you also. Thank you very much for all that you've done. Could you advance to the next slide? And I'm not gonna, go into too much detail of the data or anything, but just to kind of show what this kind of looks like behind the scenes is one of the very first studies, and Ed mentioned this, was a mouse study. And they call these engraft studies. And basically, one of the big issues you have when you try to inject these stem cell therapies is that the stem cells don't live as long as you want them to. If you don't treat them just right, these stem cells will die fairly shortly after being injected. So the first sort of proof of concept kind of study that was performed was injecting some of these myoblasts and other muscle stem cells into the tongue of a mouse, and to see if they would stay in the tongue alive and thriving for a period of time. And you can see on the bottom panel, there's this number 12 on the left-hand side across the bottom row, and that 12 stands for 12 weeks. So this is just showing that these stem cells were able to stick around and be healthy for 12 weeks after they were injected. Next slide, please. And then the next study was to try to figure out, well, that's nice that these muscle stem cells will stick around, but could they actually have the potential to do something beneficial? And there, Jan and Peter kind of came up against a roadblock, which is there's no real good animal models of dysphagia out there. So they decided to develop one from scratch. And so this is a sheep model of dysphagia where the sheep's own muscle stem cells were injected into its tongue, and then they measured several things. So in the top two pictures, one, the green one, and then the brown one next to it, what that's showing is a special kind of stain showing that the stem cells that were injected and grafted into the tongue, it fused with the tongue and it stayed living and healthy for a period of time. More importantly though, they don't look very exciting, but these bar graphs are actually very sexy, I promise you. The white and gray one here shows that there is an increase in the size of the muscle, the myofiber diameter, when you injected these muscle stem cells. So showing not only did these muscle stem cells fuse with the tissue, it made the myofibers larger in diameter, which is a really great sign. They also measured the contraction strength of the tongue after they were treated with the stem cells. And you can see in the red bar compared to the blue bar here in the middle graph that there was a significant increase in the tongue strength just from this single injection of these muscle stem cells. And then lastly is a swallowing measure here on the right-hand side. Again, the red bar is stem cell treatment, the blue bar is the before treatment. And you can see that there's an increase in swallowing pressure in the pharyngeal area. Again, a very positive sign. And it's really these two studies and a couple other ones that served as the foundation of the proof of concept that was funded by NFOSD. And it's really this set of data that allowed Jan, who already knew some of the folks at Cook and MyoSite, to say, hey, maybe we should develop a stronger relationship and try to take this through clinical trials. And that's exactly that. And MyoSite, to their credit, said, hey, this is really exciting. We'd like to add this to our portfolio. And then they funded the phase one trial. Next slide, please. Before I talk about the phase one and how good the data looked in the phase one, I just wanted to reiterate kind of what Mike said about what the purpose of these muscle stem cells are. You can see on the left-hand side, there's this schematic of a needle injecting the muscle cell, and these little gray dots are supposed to be the muscle stem cells being injected into the tongue muscle. And then eventually those muscle stem cells infuse into the tongue muscle and make it stronger and give it a greater contractile strength and eventually a greater swallow. One of the key issues here is the fact that the cells will be injected at the base of the tongue. So this is the very far back of the tongue that you don't really see when you're looking at your own tongue in the mirror. And it's this base of the tongue that serves as one of the essential key mechanisms to an effective swallow. And I'm kind of preaching to the choir here, you know, with all the SLPs, but for some of the patients, just to kind of clarify that a little bit. One of the other things that we get questions about a little bit is some patients have asked, because you guys are not just doing one injection into my tongue, but every time you inject the cells, you're injecting my tongue multiple times. Isn't that kind of bad for my tongue because it's causing an injury? And yes and no. There's actually some thought that the reverse of that may be true. There may be some benefit to actually giving some of these micro injuries in the tongue, because that's exactly this these little micro injuries that causes the muscle to want to regenerate itself. Kind of like when you go to the gym, you lift some heavy weights. What happens when you lift those heavy weights is you're creating little micro injuries in the muscle. And what happens, you go home, you rest, you eat some food, and your muscle tries to regenerate itself a little bit, gets a little bit bigger, gets a little bit stronger. So we kind of partly expect to see that our control patients, our placebo patients who are not getting the stem cell treatment, may actually see a little bit of improvement just with this needling of the tongue. Some folks call this a prolotherapy. Next slide. And yeah, so what's taken place so far in terms of the clinical development of these muscle stem cells from MyoSite is a phase 1 study and more recently a phase 2 study. The phase 1 study has been referred to as TODY, standing for tongue dysphagia. And Revive really doesn't stand for anything. We just thought it was a cool sounding name and kind of got the point across that, you know, this is a regenerative therapeutic and hopefully trying to revive some of the life in the tongue and the patient's life as well. Quickly, a brief sort of patient profile that we're looking for is for this phase 1 and phase 2 trial are patients that have oropharyngeal cancer. They're 2 years out from their last treatment for oropharyngeal cancer, and they have mild to moderate dysphagia as a result of their cancer treatment. And we're looking to enroll about 62 patients, and we'll be enrolling about another year or so for this phase 2. And we're actively enrolling, and that's part of why we're so excited to kind of present here today, is just kind of continue to get out the word to try to increase recruitment and enrollment in the trial. We have some funds to help offset patient costs for traveling. So we have several patients coming from around the country and potentially even one international patient. They're not unlimited funds for the patients to travel, but it's a decent amount per on-site visit. One last thing that, you know, kind of gets brought up to some degree for this phase 2, again, that, you know, we're having this big push on recruitment for is, you know, I want to make sure I get the stem cells, doc. You know, it's the way the trial is constructed is it's randomized and it's placebo controlled. So kind of a computer algorithm randomly picks when a patient's enrolled, do they go to the placebo arm where they don't get the stem cells, or if they get the treatment arm where they do get the stem cells. And we don't have any control of, you know, which group the patients go into. And so, but one thing that MyoSite has done that's been great to help us out with that is to ensure that the patients in the placebo arm that initially do not get their stem cells will get their stem cells once they've been unblinded at the end of their participation in the study, and they'll get those cells for free at no cost, which is really great. And this little schematic in the middle with, you know, this little stem cell, you know, life cycle, if you will, Mike kind of went over that a little bit. Basically, the stem cells that we're injecting into the tongue, they're not quite muscle cells, but that's what they're designed to do, is to turn into muscle. That's you know, they're designed to regenerate the muscle tissue. And as Mike said, the biopsy is fairly small, so you can see a picture of the biopsy that the patients get in their thigh on the bottom. It's about the size of a little grain of rice or so. this little skeletal piece of skeletal muscle gets shipped to MyoSite. They manufacture up and grow up a lot of these muscle stem cells. They freeze them down into a little vial, and then they ship it these stem cells back to us frozen. Then the doctor, either Peter Belafsky here at UC Davis or Clark Rosen at UCSF, which is our sister site, that's also enrolling patients. They thaw the stem cells and inject them multiple times into the patient's tongue. And this is done in 2 separate sessions that are spaced about 4 to 6 weeks apart, and receiving about a total of 300 million stem cells injected into their tongue. Next slide, please. So this is some, the title is actually slightly incorrect here. I went a little quick when I constructed this. This is actually the data from the phase 1 trial. So we're just about done, completed with the phase 1, but the data was looking so good and appeared fairly safe that we were allowed to go ahead and start the phase 2 as we're kind of winding down the phase 1. An interesting thing, you know, on the clinical side of things is, how does one measure success? You know, what are the validated measures that one could use in a clinical trial to validate that these stem cells are indeed having, you know, a positive benefit? And you know, when we have that discussion, a lot of times it's around this technical term of validated measures. You know, what's already gone through clinical trials and shown to be a good measure. Well, let's use that measure again. And the problem that we face is nothing like this has ever been performed before in a clinical trial, so nobody really knows what the validated measures are. What's the key metric that we should measure in the patients that will, you know, give the best indication of a positive signal or positive benefit in the patients. And so as a consequence of that, we're measuring a number of different things. You kind of have to pick what your primary measure is going to be, and for us, that's the IOP device. For those of you not familiar, the IOP device is just a little balloon that gets placed on the top of the tongue. The patient squeezes the balloon with the tongue as hard as they can, and the IOP device measures their tongue strength. And what we can see in this primary measurement for the phase 1 study is that the patients showed an increase in their tongue strength. So as we all know, dysphagia is progressive and the patient should be getting worse over time. So even just keeping the patients at the same level over time would be a big win, but the therapy didn't seem to keep the patient's steady state. You know, a year after we measured them, it actually increased their tongue strength, which is great to see. One little nuance about the phase 1 is they tested 2 different doses, a low dose and a high dose. The low dose was 150 million cells, the high dose was 300 million cells. And this data in the middle graph is combining both of those together. But if you look at the graph on the right-hand side, if you tease those apart, the low dose and the high dose, you see a trend, a non-statistically significant trend in the higher dose having a greater effect on increasing the tongue strength. Phase 1 studies, there aren't that many patients enrolled in them. Normally for our study, it was about 20 patients. And so you're not really statistically powered to look at efficacy. The main thing you want to really focus on is safety. So it's not really fair to make too strong a judgment on how well this potential product works or not in these dysphagia patients. But the data nonetheless looks very, very promising, and that's partly how we were able to convince CIRM to give us a large $11 million grant to perform this phase 2 that we're doing now. Next slide. Did it skip a slide by any chance? No, okay, sorry. So just a rough structure, I've talked about some of this, is it's a double-blinded placebo-controlled trial. So the investigator and also the people interacting with the patients are not aware of who's receiving the stem cells or the placebo. And that's to avoid any sort of bias being introduced. The placebo-controlled part of that means that some of the patients will be given the stem cell therapy, other patients will be just given a placebo that's inert, and half the patients will get one, the other half will get the other. And that's so we can kind of do a head-to-head comparison under the same conditions, under the same procedures, with the same doctors, you know, really testing whether the stem cells under those control conditions work well. And I believe I talked about most of this. I'd also like to just give a big shout out to Peter Belafsky here at UC Davis, which is where I work, and also Clark Rosen at UCSF, and Mike at MyoSite, and all the people at CERM. There's actually a big team of people at each of these sites, and it's impossible to mention everybody. We'd be here for like 15 minutes just listing off names that we don't, but it's as sort of the program manager for this trial, it's been really great to interact with these groups because everyone has a very gung-ho, proactive, friendly, and cordial approach to thing, and always thinking about putting the patients first. And it's just been a very awesome experience to work with everyone on the trial so far. So yeah, with that, I'll kind of wind it down. It looks like we're starting to run out of time, and happy to take any questions you may have. Oh yeah, thank you for putting that up. Sorry about that, Mike. If you have any patients that you think may be interested, or if you want to find out more, you can scan this study website QR code, and it'll take you to a study pages site that's specific for the Revive clinical trial. It'll give you a little bit more information. It'll also ask you if you want additional information that we can send you about the trial. And then on the right-hand side, we have QR code for our intake survey. This is the quickest way to find out if a patient might potentially qualify or be eligible to be in the study. It's a quick survey. It takes about 5 minutes to fill out, if that. And then down below, we have an email address that's specific just for this one study. It's this HS-ENT stem cell at ucdavis.edu email address. You have any questions at all, would happy to address that for you. We have a whole team that responds to this email address, so we should get you good coverage on that. And I mentioned these sort of key points. We have 2 sites at UC Davis and at UCSF, but again, we're taking patients from all over the country and even internationally, where that's feasible. And the placebo patients will be offered the stem cell treatment after they've been unblinded at the end of the participation. And we have some travel funds available to help offset some of the travel burden. There's about 8 on-site visits or so throughout the course of the trial. So if they're traveling from across the country, that's a non-trivial potential financial burden on the patients, and so we were able to offset that a little bit. And yeah, just really excited about the study, and happy to answer any questions you might have.
Well, Jonathan and Michael, thank you so much for presenting this evening. I know there's a lot of people on the call in our community that are survivors of head and neck cancer, and they've really run out of treatment options. So I think this work is inspirational to them, certainly to the foundation itself. And there's a lot of people on this call also that supported that initial work. So it's nice to see their names here tonight. We do have a few questions that have come in, so I'll get started with those. Feel free to, whoever wants to respond is just fine. I'm going to start at the top here. Could you remind us what the source is for the stem cells from the patients, and kind of a quick overview again of how you procure them?
Yeah, absolutely. So the source so that we get the cells from comes from the patient's own quadricep muscle. So we take that biopsy, and I'll just explain it. But yeah, so we take that biopsy from the patient's leg, and then that muscle tissue is then shipped to our facility in Pittsburgh. So whether it's at UC Davis or UCSF, they ship the tissue from your leg to our Pittsburgh facility, where we isolate out the muscle progenitor cells from that tissue sample, expand them, and then send them back so that they're injected back into you. So your own cells are expanded, isolated and expanded, and sent back into you, or sent back to your physician for injection.
Excellent. And patients that are in the trial, whether they're in the placebo group or the actual group receiving the stem cells, both of them undergo the biopsy at the beginning, is that correct?
Correct, yeah. So they both undergo the biopsy at the beginning because it's a blinded study. So everyone needs to remain blinded and go through the same procedures. So everyone undergoes the biopsy at the beginning. But then, as Jonathan mentioned, even if you receive placebo, once you're unblinded at the end of the study, you have the opportunity to receive your cells. So at MyoSite, we will have your cells ready to go for you if you opt to receive them.
Excellent. Question for you, Michael. Do you work in conjunction with the University of Pennsylvania?
No, we do not. We are not currently working with the University of Pennsylvania, but that's something we, in particular for the study in particular. So that's not something we currently have ongoing, but it's something that we could discuss, certainly.
And Jonathan, I have a question here for you. Going back to the slides that showed one of the graphs about the outcomes, I think it was for the phase 1 that showed the increased pharyngeal pressure. Can you speak to the cause of the increased pharyngeal pressure? Was that due to increased tongue-based contraction or something else?
I can't, the hand wavy explanation is that we think it's an increase in contractile strength of the base of the tongue. The way the study was constructed, we're not able to specifically point to that being the exact cause, but it seemed it's a reasonable hypothesis.
Excellent, thank you. A question about the rationale for accepting patients into the trial that have mild to moderate dysphagia rather than severe dysphagia.
You want to take that, or you want me to take that, Mike?
You go ahead. I'll fill in.
Yeah, so there's two main axes around which the physicians were kind of wrapping their heads around in terms of what's the best shot at success here. How do we get this thing approved and show whether or not it's actually going to have a positive effect or not. The first thing we have to keep in mind is there's a bunch of different patient populations that have dysphagia. It's not just always due to head and neck cancer toxicity. It's from a stroke, a variety of different neurological disorders as well. The list kind of goes on. The thing that happens though in these clinical trials when you try to analyze the data is the more diverse the patient population is in terms of being from a variety of different diseases and different underlying mechanisms going on in those diseases, it kind of dilutes the data a little bit in terms of being able to tell did it work or not work. Because a lot of times what happens unbeknownst to us before we start the trial is that the therapy will actually work in one patient population decently well and not work so well in a different patient population. But you kind of don't know that ahead of time. So one of the things you kind of do is you hedge your bets a little bit in terms of trying to narrow a more kind of homogeneous or a singular profile of a patient population that you think might be your prime candidate to get this through. And for these trials, that's really this oropharyngeal toxicity cancer patients that have mild to moderate dysphagia. And it's also a benefit just logistically that we see a lot of those patients at both of our hospitals and our ENT clinics. On maybe a more slightly technical level, in terms of if we just keep it, with cancer toxicity induced dysphagia, is, it's the progression of two things over time. You get progressive muscle loss of muscle mass over time. So the thing you want to heal is getting smaller and smaller and smaller. If you have this really, really tiny muscle that used to be huge, but it's wasted away. So even if you double the size, you may not get a big bang for your buck in terms of functional increase with the stem cell treatment. So that's part of the thinking. The other part of the thinking with the more severe patients, which is, it's really unfortunate to kind of, talk about this stuff. I apologize, but, and again, I'd want to frame before I dive into this other thing is the goal would be is once this gets approved is to broaden the patient population of who could this apply to. You can have these off-label uses, and the idea would be to inject this into patients with more severe dysphagia for sure. But to get back to the other technical point, it's a lot of the scar tissue buildup it's kind of an issue, causes a lot of stiffness, and the greater the amount of scar tissue, the more muscle you kind of have to power through and manipulate that scar tissue. So to try to ensure if we can accurately test that this has some efficacy or not, the decision was made to look at these patients with more mild to moderate dysphagia to kind of negate those issues.
And is there a definition to mild or moderate dysphagia that you're using specifically for the trial?
Yeah, there is. There's a number of measures that we're looking at. The quickest one, I actually shouldn't say that. I'll just refer you to the study website or the intake survey, and that will kind of, it'll be very quick to fill out, and we can give you a quick response. I would say there's a couple of things that kind of seem to be like biggies in terms of eligibility criteria. one of which is the degree to which the patient's intaking their nutrition via mouth versus feeding tube. In general, we're looking for the patients to be intaking nutrition via mouth 50% or more, a little bit more than 50% of their nutrition intake by the mouth. Another thing is aspiration pneumonia. If the patient's having chronic aspiration pneumonia, that's also an exclusion criteria. That's kind of come up a few times. There's kind of a whole litany of things that are part of the, both inclusion and exclusion criteria. And I don't want to go into too much of the detail, but I would say those are a couple of the big things that kind of are limiting in terms of patients' eligibility.
Patients that have had glossectomies where a portion of the tongue has been removed, are they eligible for the study if they meet the other prerequisites?
That's a judgment call depending on how much was removed. I think in general it's around half the tongue or more needs to be present. But it's a bit of a physician judgment call in concert with all the other sort of metrics. So what this will look like is the initial stage to see if a patient is a good fit is this quick, 2-3 minute survey. They fill that out. There's an initial screen. Can we do they match any of this exclusion criteria? No. Okay, let's move them to the next step. Let's look at their medical records. We look over their medical records and we try to deep dive into those and see what we can see. And again, if everything seems to match with the intake survey, then they'll come in for a screening visit. And it's really this on-site screening visit that is the ultimate determination of whether or not they're eligible. And so they'll get swallow studies, they'll do high-resolution manometry, there'll be a physical exam, and additional assessments, including patient questionnaire, intake questionnaires. And it's in concert with all those things that the ultimate judgment of, in terms of from the physician's point of view, is this candidate going to be a good candidate for the trial or not? And are they on the, above moderate or moderate zone of dysphagia?
Jonathan, do you want me to share it? I have the next slide too, is just as kind of supplemental that schedule of events. Do you want to show that for reference or?
Sure, yeah, why not?
Okay, yeah, you kind of ate in that talking there. There's all these assessments that are done.
Yeah, so on the left hand column, you'll see the names of a bunch of different assessments that get taken at a variety of the different visits. And each of the following columns is a number of different visits that the patient goes through. I should mention not all of these visits are on site. Many of these visits are virtual visits that just occur over the phone or via video. All the ones that have little boxes around them are on-site videos. Towards the top, the red boxes and yellow boxes, those are on site. All the ones that don't have those little colored boxes around them are just virtual visits. So patients get a little shocked sometimes when they first see this schedule of events, but it is a good number. It's 8 office visits that they have to come in. So it's a little easier if they're closer, but we have a hotel right on our medical campus that's literally right next door to the clinic that they'll be going to. So it's super easy and convenient. We have discounted rates for that. And we have an account with Uber Health to help with transportation if that's needed at all. Again, we also have patient travel funds as well. But yeah, a number of measures, IOP, manometry, fluoroscopy, Cape V, didn't mention that. Yeah.
And Jonathan, I think you mentioned that there's potentially an international participant. As long as a person can travel and fund their travel internationally to get on site multiple times, would they be eligible to participate?
Correct. We have this decision-making process a lot of times comes down to the patient's point of view and if they have the financial means to do that. Again, we have some funds, but the further you live away from California, the less, the more expensive it gets to travel in, and there's a cap on how much funds we can give. It's about $800 per patient per on-site visit. So someone who's coming internationally, this gentleman is coming potentially, from Europe, and this, the finances is not limiting for this person, so they're potentially going to be able to join the study if they pass screening. But I can see that potentially being, a bit of a financial burden for others. But just on a technical level, yeah, if that's not an issue at all, would love to take patients. We had a few interested patients from Canada as well.
Okay, excellent. For the participants that are in the placebo group, afterwards, if they choose to get the stem cell treatment, if that's an option, can that also be funded in part by the study, the travel expenses?
Correct. Yes, it will be funded to the same level. Actually, I think slightly more for the follow-up arm. I think it's $1,000 per visit per patient for that sort of extension portion of the study where the placebo patients receive their cells. And there's also much less visits, so it's much less burdensome. So the patients who had to come in for those 8 on-site visits for the main part of the trial, if they choose to get their cells after they're unblinded, they just have to come in for the 2 visits for the 2 injections. And then after that, all the visits will just be video and or telephone visits, just virtual visits. So they won't have to come on site anymore after they get the stem cell injections. We'll just be following them for safety at that point.
Excellent, thank you. I have another question here for you. Can you discuss the neuropathy of the hypoglossal nerve and whether patients will be eligible if they have this type of injury? And if so, how does improved muscle bulk without proper nerve innervation improve the function?
Could you repeat that one more time? It broke up a little bit when you're...
Absolutely. So a question here about neuropathy of the hypoglossal nerve and whether patients with this type of injury would be eligible. And if they are eligible, how does improved muscle bulk work if you don't have the proper nerve innervation to improve the function?
All right, so we're going to reach some limits of my knowledge here, but I'm going to do my best to answer this question. I'm not an ENT physician. I am a research nerd that spends time in a lab working with animal models and stem cells. Having said that, this is my understanding: is it's been voiced to me by ENT experts that it's not always clear when there's a deficit in the swallowing function, to what degree that's due to nerve damage versus muscle damage versus scar tissue. And that when we say that we think it's perhaps nerve damage, there's a degree of speculation that goes along with that. So that is not an exclusion factor at all. So anybody that is thought to have nerve damage is more than welcome to come through the screening and be assessed and participate in the study for sure, because it's not always clear if that's the exact thing that's going on. And then the second point of that would, our second part of the question was, well, if you're going to bulk up and beef up the muscle, what does that matter if you don't have a nerve there to fire that muscle? Great question. And that's true, yeah. If you do have a big muscle there and you don't have nerve fibers there to trigger that muscle to fire, which is how our muscles contract, they receive a signal from the nerves to contract. And if they don't receive that, they won't contract. So what's the point? It comes down to, we're kind of over, when we're talking about the way the muscle stem cells function, we're kind of simplifying things a little bit. And one of the things that's going on that we didn't talk about is this idea of communication signals, which happens to be my specialty, my research specialty, so I can speak at some length about this. And there's this very interesting idea that cells actually literally, not figuratively, they're literally passing little packets of information back and forth to each other. This could be little proteins, it could be little small bubbles called exosome vesicles or extracellular vesicles, and they're literally little packets of information, and they use to talk to each other, and they use to trigger certain kinds of behavior in the cells and tissues surrounding them. One of the things that we know that nerves respond to is growth factors, right? So a lot of the nerves and the vascular system grow in parallel, kind of like railroad tracks. And what triggers that during development is the secretion of growth factors along the track that they're going along. Almost like adding fertilizer for them to grow out in that direction. And one of these things that we know that the stem cells do when they're injected is they secrete a lot of these growth factors. So even though that's this idea of triggering nerve growth is not the main mechanism that we think is going on here, there's definitely data that MyoSites and the founders of MyoSite have published where they show that the muscle is innervative. And you can see that sort of proof of pudding, you know, and you can even make an argument if it's the muscle's innervated, is it even going to fire? But I think the real proof of it is in the efficacy data. When you look at like, yeah, when we measure the strength, does the strength increase or does it not increase? And clearly, if it's increasing, then there's a nerve there firing that muscle. So there hasn't been as much data generated about this idea of the muscle stem cells potentially inducing a higher degree of innervation. It does happen to some degree. The main mechanism is thought to be through bulking up the muscle. But having said that, you can actually see in the efficacy data that the muscle does contract stronger. So it would lend itself to thinking that it gets appropriately innervated along the way.
And I will just add just one quick note. I know we're over 8, but the level of neuropathy will be assessed by the physician and up to the physician's discretion if it's a good fit for the study. Up to Jonathan's point from earlier in the Q&A, really one of the goals of the study design is create as homogeneous of a patient population as possible. So trying not to bring in too many factors into too many variables into the situation. So it'll be something that is discussed and assessed during the assessment.
All right. Jonathan and Michael, I want to be mindful of your time tonight, but I do want to end with just one more question here before we wrap things up. My last question for you, are there any long-term problems or possible consequences of undergoing the stem cell treatment? For example, is there an increased risk of recurrence of cancer because there's increased tongue muscle, perhaps?
I could speak to that.
Sure.
So I could talk to what we've seen happen in the 20 or so patients that have been injected to date. The two main issues, one, it seems fairly safe overall in a nutshell. Again, it's in a small number of patients that have only been followed for a certain period of time. We need to do more patients, follow them out for longer. That's what the phase 2 and phase 3 trials are for. But so far, so good. Occasionally a patient, when they get the biopsy out of their thigh, I think we've had one hematoma develop in the thigh. It resolved. It wasn't an ongoing issue. And then there's some degree of inflammation that happens following the injection. You know, there's a number of needle sticks in the tongue, so that causes some injury and a little bit of inflammation and some discomfort associated with that. But so far we haven't had any severe adverse events that have been related to the procedures or treatments in of themselves. And could you repeat the question? I had like one other point, but I just spaced on what it was.
I think the last part of that question was if there was an increased risk for cancer recurrence.
Oh yeah. This is a great question, and it takes a little bit of a historic understanding to answer that question. The FDA, when you go to propose using a new type of therapy, is super risk averse, and it's safety, 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 safety. Safety is always first. And something you might have insinuated correctly, I would add, from Mike's slide is that, hey, stem cells like to proliferate. So if we're injecting stem cells and they like to proliferate, does that start to sound like cancer? You know, what's cancer? Cancer is a group of cells that have uncontrolled proliferation, uncontrolled cell divisions, right? And so that's from the history of stem cell research, which started back in the 50s. And incidentally, the first stem cell treatment has already been approved. It's called a bone marrow transplant. And that's been approved for a number of decades. The millionth patient has been treated with that successfully. But it is a big concern, and that's and historically, when the way the FDA has approached that is from a very cautious point of view, which is they wanted the patients to be quite a few number of years post cancer treatment to make sure that their cancer wasn't coming back. And the idea that you didn't want to put stem cells in there that were dividing and secreting growth factors to make that recurrent tumor worse. The other part of that is they want to follow, they had originally for a lot of the original stem cell trials, they were wanting to follow the patients out for a really long time. And the idea there is, okay, well, maybe there wasn't a recurrent tumor, but maybe the stem cells themselves will cause a tumor. And so they were having the clinicians follow the patients out, in some cases out for a decade, you know, was what the FDA was wanting folks to do, which is really challenging. But what's kind of happened over the intervening decades is that it's been kind of recognized for most of these stem cell therapies that that's not a concern that's been materialized. Thousands of patients have been injected. This hasn't been seen a single time, i.e. inducing a tumor. That's not to say it couldn't possibly happen at some point, but as we kind of understand the mechanisms of how these cells work and how they don't work, and what the data actually has come from the trials, there hasn't been a single case of a human getting cancer from stem cell treatment. Again, thousands of patients have been injected at this point.
And I will add, just to specifically for this product and this technology of autologous muscle-derived cells, not only the tongue dysphagia indication, but the other 3 programs we have for our clinical trials. Across all 4 of those programs, we have treated over 700 individuals with this technology, and it's a very safe. It's a very safe, very nice safety profile with that. Jonathan touched on really some of the major things that pop up. Most of them are related to the biopsy procedure, just with procedural pain, and then also injection site inflammation. But for the actual product, we've seen a great safety profile.
It's really helpful and hopeful to hear. Thank you for sharing that. It's about 10 minutes past the top of the hour, so I will wrap things up. There were some questions that came in that we didn't have time to get to. If you had a question that wasn't answered and you would like to know the answer to that, please feel free to email us. We will be sending out an email after this webinar that will include our email address, but also some links and PDFs so that you can have more information about this clinical trial. And again, you'll have the email to contact Jonathan too if you're interested in more information about the trial. Michael and Jonathan, thank you both for sharing this information with our community tonight. We really appreciate it. I'm going to invite Ed to join us to wrap things up here. So Ed, go ahead and take it over.
Jonathan, Michael, and you did an amazing job describing the work you're doing, and I hope that the therapists and the patients on the call tonight that think they have an interest will follow up with you and assist with the recruitment of patients for this trial. So I want to thank both of you. I also want to thank Elizabeth Daly, who is the Foundation's Executive Director, for facilitating the questions this evening. And again, I really enjoyed the tour of a world-class facility. So thank you both. And with that, I think we're going to sign off. Good night, everyone.
Thank you, everybody.
Not everyone. Thank you.